Good day, viewers. Today we are going to see about an interesting topic, colorimetry, its principles, instrumentation, and application. Measuring colors, understanding life, quantifying biochemistry with precision is a motto of colorimetry. And in the series of lectures, we will be discussing about different molecules estimated using the principle of colorimetry in clinical biochemistry practices. Coming to colorimetry. Colorimetry is an analytical technique used to determine the concentration of colored substance in a solution by measuring the intensity of color produced. In simple words, it is measurement of absorbance or transmittance of visible light by a controlled solution, colored solution. You might have a question in your mind, why colorimetry when we have other instrumentations? Because it is simple, rapid, reliable and cost effective, requires very small volume, which is suitable for routine laboratory analysis and is widely used in clinical, biochemical, pharmaceutical, food and environmental analysis. Coming to the principle of colorimetry, it is based on the measurement of absorbance or transmittance of visible light by a colored solution. The intensity of the color developed is directly proportional to the concentration of the substance in the solution. It is given by Beer-Lambert's law. When monochromatic light passes through a solution, absorbance is directly proportional to the concentration of the absorbing species and the path length, which is given by the equation A is equal to epsilon Bc, where A is absorbance and epsilon is a molar absorptivity and B is a path length, which is always kept as 1 centimeter and it is constant. And the C is a variable, which is the concentration of the particular biomolecule in the solution. In the ideal conditions for the colorimetry are, it should have a monochromatic light. A light with single wavelength should be used. Solution should be clear and it should be free from turbidity. No chemical changes should occur between analyte and the reagent with the time of measurement. QH should be clean and of fixed path length. Measurement should be made at the wavelength of maximum absorbance of the greater sensitivity. The wavelength selection can be done for each substances where it shows maximum absorbance at a particular wavelength, which is called as lambda max. At this particular wavelength, the substance will have high sensitivity and greater accuracy. Upon plotting the concentration with the absorbance, we will have a linear graph based on which we can extrapolate the unknown concentration present in the solution with the help of the standards. Coming to the instrumentation, a colorimetry measures the intensity of light absorbed by a colored solution. The instrument consists of optical and electronic components arranged to measure the absorbance or transmittance. The optical path is as follows. Light source enters your lens and it is concentrated in a monochromator. Then with the help of wavelength selector, it is selected and it is allowed to pass through the sample in the cuvette. The absorbance or the transmittance is detected by the detector in the photocell and the readout it is read out in the display unit as either absorbance or the transmittance. Coming to the components, light source provides light. The colorimeter lens converts the diverging light from the source into a parallel beam. Then the monochromator disperses the polychromatic light and isolates a desired wavelength. The wavelength selector slit allows only a narrow band of wavelength to pass through. And in the sample, in the cuvette, the sample holder holds the sample. The detector, which is there as a photocell or photoelectro cell, converts the light energy into electrical signal. And the readout or display device shows the reading in terms of absorbance or transmittance. We have various types of colorimeter based on the filter, based on like photoelectric colorimeter, digital colorimeter, spectrophotometer, etc. Coming to the clinical applications of colorimetry, it is widely used in clinical biochemistry to estimate various biomolecules such as blood glucose, urea, creatinine, total protein, bilirubin. Apart from this is also used in estimation of cholesterol, triglycerides, uric acid, and electrolytes like calcium, phosphorus, iron, and it also estimates the amount of HDL to LDL, cholesterol ratio, and various enzyme assays like AST, ALP, LDH can also be performed colorimetrically. It is simple and easy to perform. It is rapid, cost effective, requires small volume, highly sensitive, and it is accurate. And it is suitable for clinical laboratory analysis. It is used in hospitals, clinical biochemistry labs, and disease diagnostic monitoring and screening. Thank you for your patient listening. Measurement today gives a better healthcare tomorrow, which is accurate, rapid, reliable, and cost effective. We will be continuing to learn quantification of different biomolecules separately in the next upcoming videos. Await for the videos. Thank you for your patient listening.